Welcome to this explainer. Today, we're totally changing how you look at a simple glass of milk forever. I know, most of us just see that plain, white liquid splashing over our cereal or into our morning coffee, right? But honestly, we're about to uncover a wildly bustling, completely invisible city of tiny biological machines living right inside your glass.
Okay, so what exactly are these machines? Well, they're called enzymes. Think of an enzyme as a tiny biological worker naturally put into the milk by the cow, specifically to speed up certain chemical reactions. There's actually around 50 of these different enzymatic workers in there, and every single one has a very specific, highly specialized day job, stuff like destroying harmful bacteria or totally changing the milk's flavor profile.
To make sense of all this, here is our master map for today. We've organized this complex chemical soup by their functional families. We'll meet the tiny machines, map out the protectors, check in with the quality checkers, confront the transformers, and finally, look at the absolute magic of making cheese.
Section 1. Meet your milk's tiny machines, a biological city.
Let's take a quick bird's-eye view of our microscopic dairy city. You've got this incredible variety of enzymes, things like lactoperoxidase, catalase, lipase, and lysozyme, all operating at wildly different activity levels. Now, I know that sounds like a bunch of heavy scientific jargon, but don't worry at all. We're going to ditch the textbook talk and translate this into basic English using some super simple mind maps.
Section 2. Mind map. The protectors, defending the milk.
We're heading into our first neighborhood right now, the natural security system of the milk. Let's build our first mind map for a protector called lactoperoxidase. Honestly, just think of this guy as the ultimate security guard. It's a natural glycoprotein packed with iron, and its one and only job is to be a powerhouse antimicrobial agent, keeping the milk safe from outside invaders.
Branching out on our map here, let's look at the stats. You've got about 60 milligrams of this per liter in cow's milk. What it does is oxidize a compound called thiocyanate, which literally stops harmful bacteria in its tracks. But here's the really cool part. This security guard has a massive superpower. It's incredibly heat stable. It actually survives the standard pasteurization process, meaning it just hangs around actively battling bacteria to extend your milk's shelf life. Now check out this crazy comparison for our next protector, lysozyme. This one is basically a microscopic wall breaker. It physically attacks and bursts bacterial cell walls. But look at this difference. Human breast milk is absolutely loaded with it. We're talking roughly 400 milligrams per liter. But cow's milk? It's barely even there, only about 0.1 milligrams per liter in trace amounts. It's just wild to see how differently these species naturally protect their babies.
Let's map out our final protector, catalase. This is a hemoprotein enzyme that spends its day breaking down hydrogen peroxide into harmless water and inactive oxygen. But for dairy farmers, it acts as a brilliant built-in sickness sensor for the cow.
So how does that work? Well, catalase levels closely parallel the white blood cell count in the milk. If a cow gets an udder infection, like mastitis, white blood cells rush in to fight it off, and they bring sky-high levels of catalase right along with them. It's a perfect natural alarm system. But unlike our tough security guard from earlier, catalase has a strict kryptonite. It gets completely wiped out if you heat it to exactly 70 degrees Celsius for 1 hour.
Section 3. Mind map. The quality checkers, proving it's safe.
Moving right along to the tiny machines used by dairy factories to guarantee the milk you buy is completely safe to drink. The core of this mind map is alkaline phosphatase, or ALP for short. Structurally, it doesn't even have any natural phosphorus or carbohydrates. But functionally, factory workers rely on ALP like an invisible thermometer.
Here's why that is so clever. We pasteurize milk to destroy really dangerous bugs, like the bacteria that causes tuberculosis. But how do you actually prove the milk got hot enough inside those massive factory pipes? Well, ALP's kryptonite is that it dies at the precise temperature required to pasteurize milk. So factories don't actually have to go hunting for tuberculosis, they just test to see if this enzyme is missing. If the ALP is dead, you know for sure the dangerous bugs are dead too.
And speaking of temperatures, 37 degrees Celsius, which happens to be human body temperature, is the exact sweet spot, the optimal operating environment for lab technicians to test this enzyme. It's the perfect temperature to check if even a tiny fraction of ALP somehow managed to survive the heating process.
Section 4. Mind map. The transformers, spoilers, and saboteurs.
Okay, we're entering a slightly darker neighborhood in our biological city. These are the enzymes that will totally ruin your milk if left to their own devices. Meet our first saboteur, plasmin. It's a proteinase, meaning its job is breaking down proteins. And get this, it literally sneaks straight out of the cow's bloodstream and directly into the milk.
Once it's in there, plasmin goes rogue, aggressively attacking and cutting up specific milk proteins, primarily beta-caseins. You know, if you've ever opened a really old carton of UHT milk and found it all gooey, thick, and bitter, yeah, that's plasmin at work. But here is the weirdest fact about it. If you try to kill plasmin with a quick, intense blast of heat, like 72 degrees Celsius for 15 seconds, you actually just destroy the biological inhibitors that were keeping it in check. So you literally make the saboteurs stronger and more active.
Next on our transformer map is lipase. If plasmin ruins the texture, lipase is your ultimate flavor spoiler. It lives bound tightly to the fat globule membranes inside the milk. Its main gig is freeing up fatty acids. The problem? Free fatty acids in milk taste absolutely awful. They cause these gross, rancid, soapy off flavors. Normally, lipase is fast asleep, but notice the activators here. If you treat raw milk too roughly, like aggressively shaking it, homogenizing it, or heating it way too fast, lipase wakes up and goes to town chewing through those fats. Luckily, its kryptonite is acid. Drop the pH to 4.6 for 4 hours and lipase is reliably stopped in its tracks.
Section 5. The Magic of Making Cheese. The Ultimate Transformation.
For our final stop, let's look at the absolute climax of dairy chemistry. We've seen enzymes that protect and spoil, but the undisputed king of deliberate transformation is an enzyme called renin, also known in the industry as chymosin. Just think of renin as a biological cheese wizard. It's a highly structured protein that thrives in acidic environments, and it is the absolute key to making almost all artisanal and commercial cheeses.
Renin basically casts a brilliant two-part spell on the milk. Step 1 is the enzymatic cut. Normally, milk proteins float around happily because they have these protective, water-loving tails made of capsaicin. Renin swoops in, acts like molecular scissors, and gives those proteins a very specific haircut, snipping off those tails. Step 2 is the physical clump. Suddenly, those proteins are unstable and scared of the water, so they grab onto free calcium ions floating by. That calcium acts like glue, cross-linking everything into a solid gel matrix. And boom, you've just made cheese curds. So, we've covered a ton of ground today. The main thing you should take away from this explainer is that milk is definitely not just some boring white liquid. It's a wildly complex, dynamic, living ecosystem. Literally, every time you pour a glass, you're interacting with a biological city of microscopic protectors, checkers, and transformers, all working around the clock.
Which really leaves us with a fascinating final thought to chew on. We know pasteurization is absolutely crucial to protect us from deadly diseases, but as we saw in our mind maps, that same intense heat wipes out some incredibly beneficial microscopic machines like catalase. It kind of makes you wonder, what other hidden biological ecosystems are we unknowingly altering, suppressing, or completely destroying in our everyday foods before they even make it to our fridge?